Welcome to my biochemistry lecture. Let's talk about structure of protein. So, the proteins are primary, secondary, tertiary and quaternary structure. Let's talk about each of them. Primary structure of protein. The primary structure of protein is a linear sequence of amino acid. In protein, the linear structure of amino acid is either 3-letter or 1-letter form. There are the amino acids are attached by each other by the peptide bonds. Now, the primary structure of protein is always written at the C-terminal end or first to the N-terminal end to C-terminal end of the last amino acids. The information of primary structure of protein can be obtained by sequencing the gene that encode the protein. The information cannot be obtained by sequencing the protein itself. Secondary structure of protein. The primary structure of protein, the amino acids are linked by the peptides bond. In secondary, the peptides bonds of the like oxygen attached to the carbon and also the nitrogen attached to the hydrogen. Both the oxygen and also hydrogen have ability to form the hydrogen bond. The result effect in the alpha helix as well as the beta sheets. We will discuss about the alpha helix. Alpha helix is most common secondary structure. In this structure, protein peptides are in spiral structure or spiral in shape, such that the side chain of amino acid unfold and alpha helix is stabilized by the hydrogen bonding. The hydrogen bonding again form the hydrogen, nitrogen and hydrogen oxygen attached to the common carbon. Individual hydrogen bonding is weak, but collectively it establishes the spiral structure. Peptide bond, except the first and last, participate in the hydrogen bonding. Each tier of the helix is all about 3.6 amino acid and the distance between them is 0.54 nanometer. Presence of amino acid proline makes the helix is unstable. Protein. Beta sheets. Beta sheets is the secondary structure of protein. Now in the alpha helix, hydrogen bonds in between the peptide chain, while in the beta sheets, the hydrogen bond is formed between the different peptides of the polypeptide chain. It can be parallel or it can be antiparallel infection. Now come to the tertiary structure of protein. The tertiary structure of the protein refers to the overall three-dimensional arrangement of its polypeptide chain in space. The protein structure are made by condensation of amino acid forming the peptide bonds. The sequence of amino acid in the protein is called as the primary structure. The secondary structure is determined by the dihedral angles of the peptide bonds. The tertiary structure by the folding of the protein chain in the space is formed. It is generally established by polar hydrogen and ionic bond interaction, internal hydrophobic interaction between non-polar amino acid side chains. Come to the quaternary structure of protein. Tertiary structure of protein further interact with the other tertiary structure of protein to form the dimer, trimer and also tetramer. the tertiary structure and also the quaternary structure of the protein found in the most of the cells. Thank you for watching.